Yeah, thanks, Tom. You haven't met me yet. I'm Surein. Very good. You've seen me a few times. I thought I was going to have a break, but thank you for including me again in another session. So I'm actually presenting a study that's completed recruitment called the SHERLOCK study. So actually, Ben gave a fantastic talk yesterday about all the different changes in KRAS G12C mutant lung cancer. And by way of background, this study obviously started before we had next generation KRAS inhibitors. So, you know, one could argue where does sotorasib sit in the landscape? Nevertheless, this is a study that we presented at World Lung last year. So sotorasib, first-in-class KRAS G12C inhibitor. We saw that in the second-line setting, it beat docetaxel in progression-free survival, available in the US and in Europe, but not available in Australia. So we wanted to examine it in the first-line setting based on some data that was initially done by the Japanese, the SCARLET study, where they showed a fantastic response rate, up to 89%, using this combination of chemotherapy with sotorasib. And the reason why at the time we didn't include immunotherapy is because sotorasib and immunotherapy, PD-1 drugs, are very toxic. So hence we went down with this approach, but we used bevacizumab instead. So 52 patients were recruited to the study, first-line KRAS G12C mutant non-small cell lung cancer, and patients with asymptomatic untreated brain metastases were allowed. It was single-arm phase 2. Patients received the platinum, pemetrexed, bevacizumab biosimilar, and sotorasib, 4 cycles followed by maintenance, and the primary endpoint was objective response rate, including key secondary endpoints such as progression-free survival, duration of response, depth of response, overall survival, adverse events, and there was some correlative analyses that we are currently planning and in negotiation for. So first of all, I wanted to use this opportunity to thank all the primary investigators, sub-investigators, study coordinators, and of course the patients who made this study happen. So thank you very much. We recruited across 13 sites. And as I mentioned, Chi, who is currently on a flight to Malaysia via Singapore, quickly wanted to fly Singapore Airlines, is currently, well, he presented at World Lung last year. So the baseline characteristics for the study, you can see the median age was 67, 62% were female. As expected, the vast majority had a smoking history, and 23% of patients had a high PD-L1. And we'll talk about that because naturally, I'm sure you're thinking in your heads, really, if you're high PD-L1, why should we go with an approach that doesn't include a checkpoint inhibitor? We also know that concomitant mutations with KRAS mutations may be, and we believe, are predictive biomarkers in the sense that they diminish the potential impact from these checkpoint inhibitors. So STK11, 32%, and KEAP1, 18%, is very consistent with what we've seen in the literature across other studies with KRAS G12C mutations as well. So this data will, of course, need to be updated. So we are sort of a year since this initial presentation. And Deborah, that you heard earlier, is working very hard on putting together the latest figures because we are about to put together the final manuscript, which is currently in draft. So here you can see the confirmed objective response rate was 64%, and the unconfirmed objective response rate was 69%. And in the spider plot, you can see there were durable responses up to 21 months. And in fact, I can also share that at this stage, 12 patients remain on the study, with some of the patients now over 3 years. So some patients truly derive excellent benefit from a immunotherapy-free strategy. Here you can see, and somewhat surprisingly, and I know these numbers are small, and I would still argue hypothesis generating, but nevertheless, the patients with PD-L1 over 50% actually had the best response rate, so up to 80%. And then you can see that PD-L1 negative, about 57%. Importantly, this benefit was seen across STK11 and KEAP1 subgroups. So that's a very important finding as well. The median progression-free survival, 9.1 months. Median overall survival, 25 months. This data will be updated prior to the final publication. And as I mentioned, we looked at the concomitant mutations, whether it was TP53, but really KEAP1 STK11, we observed that there was fairly similar progression-free survival outcomes as well. Naturally, when you add bevacizumab to these in a 4-drug combination, what are the toxicities? There you can see that 23% of patients on sotorasib had to discontinue, 13% of bevacizumab had to discontinue, and then there were a percentage of patients in a dose reduction. 5 patients died while on the trial, and 1 had a bronchopulmonary hemorrhage, and we amended the protocol to be very strict about anticoagulation use, because that particular patient was on anticoagulation going to the trial. There you can see the toxicity rates. Vast majority were grade 1, grade 2, mostly chemotherapy-based toxicities, and generally, as I said, well-managed with supportive care medications. So conclusion, sotorasib, bevacizumab, and chemotherapy demonstrated promising anti-tumor activity in the first-line setting of KRAS G12C mutant non-small cell lung cancer. We observed high objective response rates across all PD-L1 subgroups, but particularly the PD-L1 over 50% expressing subgroup, and that appeared to work across KEAP1 STK11 mutations. It demonstrated a manageable safety profile. And I'm not sure whether it warrants larger trials to evaluate that particular combination, but I think it definitely gives us some key insights into how potential next-generation KRAS inhibitor studies should be used. And to my knowledge, anti-VEGF therapy is not part of that. But of course, we are seeing a lot of work that Sanjay discussed yesterday with PD-1 VEGF strategies. So maybe the next sort of iteration of trial should be a next-generation KRAS inhibitor plus a bispecific PD-1/VEGF, and maybe we can chuck in some CTLA-4, we get a fantastic combination. Thank you very much.
Fabulous, Surein. Well done. I mean, I think this is an excellent study and we delivered so well. So you answered my question I was going to ask, of course, which was the future direction, and I agree. I mean, maybe not with the 4-drug combination, but that would be a little bit exciting, wouldn't it? It's a busy space, KRAS. Do you think we can do another IIT and move forward with the bispecific next gen?
It is a busy space, and of course, it's very, as Ben showed you, I don't know how many KRAS inhibitors are out there. You can't even count them. He didn't even have the complete slide. So there's a lot, and it's very pharma-driven at the moment. And I think that we're going to have to think through this as a TOGA committee and how best we can potentially impact this. And sometimes being the right place at the right time is important. How we collaborate. I think that I do believe that once we see the readouts from the KRAS inhibitor plus the Pembro trials, the next natural question, and we're going to see Rosetta Lung 02 and these sorts of studies read out. So we're going to have these 2 hopefully positive trials, and I think it will make perfect sense to use a KRAS inhibitor with pumitamig or ivonescimab or all the other combinations like that.
Thanks, Surein. Can I just ask a question about, so it's quite intriguing that PD-L1 greater than 50% doing so well. So just sort of 2 aspects. Firstly, why do you think they had much better response rates? And second, have you followed what's happened to that group of patients? Do they, presumably if they go on to immunotherapy, they also have very good outcomes?
Yes, that's a good question that is currently being worked through. Deborah is putting it all together, and I think she's also capturing next line and what happens with immunotherapy. Not quite sure what's happening with the high PD-L1. That was quite a surprising finding, I have to say. But we're currently trying to work out some translational signs. So we're hoping to, basically do some whole exome sequencing and try to do some spatial transcriptomics, etc. So we're currently negotiating in terms of the best way to do that. And at the moment, because as Chi said to me, his expectation was that most of the patients be off the trial by now, but there's still plenty of patients on, so we haven't retrieved the tissue and the sampling from all the various hospitals. So we're trying to work out at what point do we draw the line in terms of actually collecting the tissue. And we're thinking that we probably should, because some of these patients who are on trial, once you've been on it for 3 years, what happens after that? I mean, do you just keep going? Like, we don't quite know how this is going to play out.
And remind me, you collected liquid longitudinally, and is there a plan to sort of interrogate the results there? Is there funding to do that, looking at this question?
So we have done Foundation 1 and the baseline, I think I can't remember what it was, and at the 9-week mark as well. So we do have those results, and there's progression bloods as well. So we want to kind of have a look at all that as well.
Okay, thanks, Surein.